Almost everyone pictures CRISPR the same way: a tiny pair of molecular scissors that snips out a broken gene and drops a healthy one into the gap. That picture is wrong. What CRISPR actually does is stranger, and it began as a weapon bacteria built to fight viruses, a search engine that hunts through 3 billion letters of your DNA to find one exact target. To see why it works, look at where it comes from. Bacteria get infected by viruses too, and they have fought them for billions of years. When a virus injects its DNA and the bacterium survives, it grabs a small piece of that viral DNA and files it inside its own genome, like keeping a mugshot of an attacker. If that virus ever returns, the bacterium copies the mugshot into a short strip of RNA, clips it into a patrolling protein, and sends the pair out to shred any DNA that matches. That protein is Cas9. That strip of RNA is the guide. Here is the move that turned a bacterial defense into a tool. That guide strip is just a sequence of letters. Swap in a different sequence, and the same protein goes looking for whatever you tell it to. Jennifer Doudna and Emmanuel Charpentier showed you could do exactly that and won the 2020 Nobel Prize for it. The press called them genetic scissors, and that name is where the trouble starts, because cutting is only half the story. So watch what actually happens at the target. The guide carries a search string about 20 letters long, but Cas9 does not read the whole genome letter by letter. That would take forever. Instead, it races along the DNA, feeling for a tiny flag, a tag just a few letters long that sits next to real targets. Only when it finds that flag does it stop and check whether the 20 letters next door match its guide. If they do, Cas9 pries the helix apart. The guide zips onto one strand, and 2 molecular blades cut both strands, about 3 letters from the flag. And here is the honest twist the scissors name hides. Cas9 makes the cut, then it lets go. It never puts anything back. The actual editing is done by the cell itself, scrambling to repair a break in its own DNA. Usually, the cell just jams the broken ends together, sloppily, losing or adding a few letters at the seam. And that mangled seam breaks whatever gene was there. So first-generation CRISPR is fantastic at switching a gene off and clumsy at fixing one. To rewrite a gene precisely, you have to hand the cell a template and hope it copies from it. And most cells will not. So if breaking genes is easy and rewriting them is hard, how do you fix a single wrong letter without smashing the DNA to get there? That is the real precision revolution, and it is quieter than a cut. The first answer is a base editor. Think of it as a molecular pencil. You take a Cas9, disarmed so it can no longer cut both strands, and bolt a chemical tool onto it. It still uses a guide to park on the exact spot, but instead of slicing, it reaches in and chemically rewrites one letter into another, an A into a G, say, leaving the DNA backbone intact. The second answer goes further. A prime editor works like search and replace. It nicks just one strand, then uses its guide as both an address and a tiny scrap of written instructions, copying short new text straight into the DNA. It can make edits base editors cannot, though it is newer and still early in the clinic. Together, these tools flipped the goal, from where to cut to which letter to write. This is not a thought experiment anymore. A little boy named KJ Muldoon is why. He was born with a broken gene called CPS1, a mistake so rare it strikes about one baby in a million. His liver could not clear ammonia from his blood, a poison that builds up from ordinary food and can wreck the brain. Roughly half of these babies die in infancy, and he was too fragile to survive the wait for a transplant. So a team in Philadelphia did something no one had done for a single patient. They read KJ's exact misspelled letter and built a base editor aimed at his mutation alone. Then, they had to get it into his liver cells while he was alive. They wrapped the editor in microscopic bubbles of fat, lipid nanoparticles, and infused them into his bloodstream. Those bubbles drift naturally to the liver, slip inside the cells, and release the editor to find his mutation and flip it. He got 3 infusions in the spring of 2025, and it worked well enough to matter. He could eat more normal protein, needed less medication, and rode out colds that used to be emergencies. After 307 days, he went home. But be careful with the word cure. KJ's own doctors will not use it. A deadly disease was turned into a much milder one, and he is still watched closely. A year later, he was walking and talking, and he went to Washington with his family and medical team to argue that he should be the first of many. It is one of the most hopeful things medicine has done recently, and still an experiment with a single precious data point. KJ was one child. To see CRISPR at real scale, look at the one that is already approved medicine, Casgevy, for sickle-cell disease, an inherited flaw that warps red blood cells into stiff crescents that jam blood vessels and cause brutal pain. It does not go after the broken gene directly. Instead, it flips a hidden backup switch. Everyone is born making a special fetal version of hemoglobin, then shuts it off after birth. Casgevy uses CRISPR to disable that off switch, so the body makes fetal hemoglobin again, and the rescued cells stop sickling. But here is the catch. Casgevy is not a shot. It is closer to a bone marrow transplant. Doctors pull blood stem cells out of the patient, edit them outside the body, wipe out the old marrow with chemotherapy, then put the edited cells back. That works because blood is one of the few tissues you can take out, fix, and return. The FDA approved it at the end of 2023, and on July 1, 2026, expanded it down to children as young as 2, opening it to a few thousand more kids. The price runs around $2 million per patient. So here is the honest scorecard. What is solid? Editing blood is approved, and delivering an editor to the liver has now saved a child. What is hard? Almost everywhere else. The editor is big and fragile, and getting it safely into the brain, muscle, or heart in enough cells to matter is still mostly unsolved. There is always some risk it flips a spot it should not, so every therapy is screened for that. And a treatment built for one child is a moonshot, not something you can hand to 1 million people. And there is one line all of this stays behind. Every therapy here edits a patient's own body cells, blood, liver, the tissues that are already sick. None of it touches eggs, sperm, or embryos, so none of these edits pass to a person's children. That is the bright line between fixing a patient and engineering a bloodline, the line one scientist crossed with edited embryos in 2018 and was condemned and jailed for. This medicine does not go there. The scissors cartoon was never quite right. What we actually have is stranger and more careful. A search engine borrowed from bacteria, learning to find one letter among billions and quietly rewrite it. It does not swap a bad gene for a good one on demand. It is a method that just went from one rescued child toward a plan to treat the next handful, one carefully written letter at a time.